Thank you. Welcome to session 6 of the WALPA annual conference. This time it's on invasive management, invasive species management. I'm Joan Hardy, toxicologist and secretary of WALPA. And I'd like to make 2 call outs before we start. And just, I think we had a great start to new and productive careers with those student posters. Just really want to say those were great and really enjoyed those. Also, to Tim Clark, that that project has been the most entertaining, interesting, and rewarding project in any limnology that I've done. So thank you, Tim. Okay, so to get started, our first talk is by Julian Olden. Julian.
Good afternoon, WALPA. It's my pleasure to be here. My name is Julian Olden. I'm a professor in the School of Aquatic and Fishery Sciences at the University of Washington. Today, I'm really excited about presenting some ongoing work that Rachel Fricke and I have been doing in collaboration with Spencer Wood and Dustin Martin. Now, prevention of aquatic invasive species remains a fundamental challenge. And with hundreds of millions of recreational boaters moving among water bodies across the United States, we really need to understand what these human movements look like, because ultimately invasive species piggyback on those human movements and when they're getting transmitted across the landscape. Now, we're all keenly aware that invasive species are removed through a variety of different mechanisms, from intentional releasing of bait fish after fishing, entanglement on fishing gear and on motors, as well as being attached to boats. Many of these species shown here have been moved through these vectors, and many of these are kind of species of concern here at Washington State, whether it be mussels, fish, or aquatic nuisance species. Now, understanding human behavior really gives us an idea of the likely transmission across the landscape, and ultimately this helps us guide effective implementation of prevention strategies, thus reducing the initial introduction, spread, and then the economic ecological harm that these invaders might have. Now, to date, we've modeled the potential spread of invasive species, and I would argue that modeling actually the most invasive species, that is humans, is the most appropriate way to understand or take a multi-species approach, if you will, to understanding where invasive species might be transmitted via this vector. Now, traditional approaches to understand recreational boater and angler movement are quite limited in time, space. In fact, this behavior is often inferred from sparsely conducted in-person surveys, as shown here, or mail-in surveys, or we've done a very concentrated effort like shown here on Lake Whatcom, where we do kind of creel surveys at boat launches, and we know a lot about a specific water body. So in large part, they're limited in time and space because of limited resources. They're also limited in their representation. The reality is that recreational angling, for example, is increasing with respect to female participation and also in terms of youth participation. And these youth don't interact with these common mechanisms by which we try to understand boater movement around. They're not at the other end receiving hard mail copies of surveys. So we really need to better understand what this changing demographic is even looking like in terms of how they're using water bodies, and then ultimately what that might mean for invasive species transmission. Here I'm going to argue that mobile technologies and social media really provide new and exciting opportunities to improve our understanding of angler and boater movement behaviors at scale. That is at spatial and temporal scales in which we formerly have unable to be able to get a handle on. And in fact, there's some really interesting work showing that some of this mobile data is actually quite useful in terms of estimating actual visitation. This is some work by Spencer Wood that shows photographic visitation as estimated through Flickr, provides a good correlate of actual visitation of recreational use in different areas in the US. So the objective of this work is to map human movement in networks using data from mobile fishing applications and social media. To assess, and the implications of this work is to assess the pathways and locations of future potential movements so that we can target prevention. Now, fishing apps, there's a large variety of fishing apps. And these data are actually quite useful if we can access them, because they can give us geotag locations of where anglers are and the date in which they're actually participating in these activities. It often involves both active and passive user reporting. And it gives us some opportunities for detailed user demographic data. Today, I'm going to be talking about one such technology, and this is the iBobber, and how we use this to actually track recreational fishing activity. Now, social media is another whole form. This also gives us geotag typical photographs of people recreational on water bodies. It gives us limited demographic data, but it gives us a whole wealth of spatial data in terms of where people are recreating and when. Today, I'm going to be using Flickr data. Flickr data was highly used over the last decades or so, and it gives us a really strong geographic understanding of where potential movement are between different water bodies. So iBobber, really quick, is a solar-enabled bobber. It's shown here. You can buy it on Amazon for under $100. It provides an opportunity for anglers to understand the depths and the topography of the areas in which they're fishing. But what we're interested in is that actually pings up to the cloud every 50 seconds the location of that bobber. So that gives us an understanding of where bobbers are and where anglers are and at what time. This is the global distribution of iBobber users across the landscape. You can see it's highly concentrated in more developed parts of the world, the United States, many parts of Europe, as well as in parts of Southeast Asia. Here are the distribution of iBobber activities across the United States. We're looking at basically around 20,000 different anglers which use these iBobbers over a 2-year time period. And this included about 70,000 iBobber records. The figure shown just over my shoulder there is just the angler activity through time estimated by the amount of iBobber activity. And it makes sense with increasing fishing activity in the spring and summertime, which then dissipates over the winter. So pretty impressive coverage across the United States. Here is the activity of Flickr data across the Western United States, just for the sake of time. There's a lot of data to process. This had about 60,000 users and over 200,000 different geotagged posts across different water bodies of the Western United States. And this was done over a 13-year time period. Now, our data process was multifold. First, we have all this geotagged information about Flickr users, as well as iBobber users, shown in the 2 icons there. But a lot of that activity doesn't occur on lakes, which is what we were particularly interested in. So we essentially took their location, as well as the a lake layer, national lake layer for the United States. And then we just removed all of those geotagged occurrences that did not occur on lakes. Next, we removed all of those lakes which had no information on either iBobber or Flickr data. Then we calculated the geographic distance as one would travel on roads pairwise between each lake using a Grasshopper routing API. And then we identified the lake attributes of each one of those lakes that had activity, fishing activity on it, and also the presence of invasive species using a USGS NAS database. So first, I just wanted to take a little look at the patterns of movement behavior. So overall, between Flickr and iBobber, we had data for about 10,000 water bodies, which is quite impressive. We were quite happy with that. The plot shown on your right is just the road distance on the x-axis pairwise and the percent of anglers moved. And you can see that it's highly skewed as what we'd expect, a lot of short distance movements between different water bodies. And about 50% of anglers moved less than around 33 or 43 kilometers, depending on what data source that you looked at. So again, it's what we expect, a lot of short distance movements, but also a very fat tail. And that leptokurtic tail in this distribution is what we're really interested in as well, because that gives the indication of long-term movements, long distance movements, distance that may come from the eastern part to the western part of the United States. And if we just kind of zoom in on one of my favorite parts here in the upper Midwest, it's around the Milwaukee, Madison area in Wisconsin. This just shows the actual traced movements that we did, for example, for an iBobber user shown in red, or a bunch of geotagged Flickr data where the same user we identified in multiple different lakes through time. And this shows just the movement pathways in which they were doing and hopping, if you will, between different lakes. Now, what we can do is that we can take a look at the network of user movements. So what I've done here is I'm just shown a network analysis where each one of those dots in this plot right there is a lake, and then they're connected to other lakes through movements. So this is around the 10,000 lakes that I shown earlier. And these cluster of lakes in the same color are essentially what we call distinct components or subnetworks of this network. This indicates that they're more connected to one another than others, and this suggests that the amount of angler movement between these lakes is particularly high. Now, there's a number of components you can see there, around 700 different components, but about 10 components have greater than 100 lakes. So essentially what this is doing is allowing us to kind of zoom in on particular components where there's a lot of angler movement between them, so we can have a better understanding of kind of the flux of the movements between those different lakes, and then how we actually might manage invasive species that might get moved around because of that angler movement. So I really don't want to kind of focus too much on this kind of diagram right here, but I do want to zone in on one particular subcomponent shown in the red square right there. This had greater than 100 lakes and is highly connected, and then how we might use this information moving forward. So if we zoom in on that particular network, we're actually zooming in this part of Utah. Each one of those dots, again, is the individual lake, and they're connected through those solid lines, so nodes and different spokes, if you will. And I've just highlighted some of the major lakes that we're looking at, everywhere from Great Salt Lake, Lake Utah, and Lake Powell, lake names that we're probably well aware of. So let's just take a look at this a little bit and try to understand how we might be able to understand and use this information moving forward. So let's just zoom in on two particular lakes to show how we can use some metrics of these network topology, if you will, to try to guide prevention. For example, on the left-hand side, that's Utah Lake that has a centrality value of 123 and a mean connectedness, if you will, of 16. So centrality actually tells us how important a water body is within a network based on the frequency of visitation and connectedness to other lakes. So I like to think of lakes with really high centrality as a center of a wheel, and there's many spokes radiating outward from that lake to a lot of other water bodies. So in this particular example, this has very high average centrality. That's 16, which basically suggests that it is basically highly visited and highly connected to other water bodies. The second metric I want to highlight, and I'm going to highlight it for Deer Creek Reservoir right there, is a measure of betweenness. This is a measure of the extent to which a water body lies on paths between other very centralized water bodies. So in this example, Deer Creek Reservoir right here is kind of connecting two different kind of subcomponents of this network, those related to Lake Powell and those related to Great Salt Lake. Essentially, I like to look at this as these are kind of conduits, if you will, between two other connected networks. So through a management lens, lakes like Utah Lake shown over there with high centrality, especially those that we know that have invasive species, are really great places for required boat and cleaning gears to prevent distribution of aquatic hitchhikers among lakes within that cluster. On the other hand, Deer Creek Reservoir, or lakes that have high betweenness, really serve as bridges. That is, they're kind of connectors between other highly connected networks. And these are really prime locations for gear inspections, roadside inspections, and education to try to intercept the flow of invasive species, potentially hitchhiking between one cluster or another. Real quick, these are just the attributes of visited water bodies between those that were visited versus all within the United States. And not surprisingly, the lake area of visited water bodies is higher for those that receive angling versus not, as well as the degree of urbanization. And in fact, reservoirs, interestingly not shown here, were 10 times more often to be visited compared to natural lakes, which is discouraging because previous work has shown that reservoirs can be anywhere between 5 and 20% more vulnerable to invasive species. So not only are they more visited, but they're also more likely to have the conditions that make them conducive to the establishment of invasive species. The time between trips is quite interesting because that tells us the likely survival of these non-native species. So what I've shown here is a histogram of the number of trips on the y-axis and the days between trips on the x-axis. And we know that survival, that is desiccation tolerance, varies greatly. From on the left-hand side, I've just overlaid what hydrilla is, around 16 hours, and Chinese mystery snail, that can be over 2 months, that can remain out of water. And this just shows that this is additional filter, but the large majority of movements between these water bodies occur in that sweet spot between 0 and 10 days. Sweet spot in the sense that the large majority of invasive species would be surviving moving from one water body to another, at least according to desiccation tolerance as shown here. Our current research is looking at additional fishing apps, different forms of social media, including Flickr, Twitter data, and also trying to use online fishing forums to give us an indication or multiple lines of evidence in terms of what water bodies are getting visited more often than not. We are really excited, and this is going to be the core of Rachel's graduate work, is to kind of crack open the idea of using computer vision algorithms to indicate, what images actually are indicative of fishing or boating related activities or not. This is a really important way of us wading through the whole mass of Twitter data to try to understand what might actually be linked to angling or boater activities, i.e. the pathway in which invasive species are getting moved around. And then our hope is to try to bring all of these data sources together so that we can have a better insight of movement beyond which what we have right now in terms of traditional kind of survey-based approaches with respect to boater movement and hence the likelihood of invasive species moving around. With that, I just want to thank Rachel again, and Spencer and Dustin, who was our partner at ReelSonar, the iBobber part of this. Anyone who's interested can look at this paper, which was just published last month in the journal Neobiota. It's available online free at the URL I can show below, or you can reach out to me. And this reports just the iBobber component of the study that I just reported on right now. So with that, thank you very much for your attention, and happy to take any questions.
Thank you, Julian. The scale of that work was really impressive. And I neglected to mention that we will be taking questions at the end of the session for all of the presenters. And all of the presenters in this session have been pre-recorded. So our next speaker is Teagan Ward, and she will be talking about Whatcom boat inspections. Welcome, Teagan.
Today, I'll be talking to you about the Whatcom Boat Inspection Program and our efforts to prevent the spread of aquatic invasive species in Whatcom County. Aquatic invasive species are non-native plants, animals, and pathogens that live primarily in water and are able to thrive in new environments. They're capable of causing economic loss, environmental damage, and harm to human health. Two of the freshwater species that we're most concerned about here in Whatcom County are the zebra and quagga mussel, you can see in the bottom right-hand corner here. And they are not found in Washington State yet, but they have the potential to cause a lot of really serious impacts. These mussels have a really unique adaptation that allows them to attach to hard surfaces using tiny byssal threads. This is how they were able to be transported across the United States on recreational watercraft and equipment. And in their early life stages, they're actually microscopic and found in standing water. So can also be transported in any standing water left on boats and equipment and things like that. They cause a lot of different impacts to native biodiversity, as you can see here when they're all attached to a native mussel. Also to recreational equipment when they attach to propellers and equipment on watercraft. And then the big one for us here, as you can see on the right-hand side, that is an intake pipe that has become completely clogged with mussels, and so that can cause serious impacts to our ability to provide drinking water. They can also make beaches and recreational areas really hazardous and uninviting. As you can see, these really sharp shells just completely covering these beaches along the Great Lakes. We first found the zebra and quagga mussels in the Great Lakes area in the late 1980s, and they spread really quickly throughout the Mississippi and Missouri River basins. The red dots on this map represent the zebra mussel occurrences, and the green dots represent quagga mussel occurrences. Our first sighting of these mussels in the western U.S. happened in 2007 in Lake Mead, and since that time, we've developed inspection stations across much of the western U.S. in an effort to prevent their spread to our states. And currently, we do not have these mussels in several Pacific Northwest states, so we've been relatively successful with this effort. We are part of that effort, so we have our own boat inspection program. We focus primarily on Lake Whatcom and Lake Samish, which are the two main lakes outside Bellingham in Whatcom County. Here you can see a picture of Lake Whatcom taken from Basin 3. It has a surface area of around 5,000 acres. It is an open, multiple-use lake and is the drinking water source for over 100,000 people. The Lake Whatcom watershed is also home to around 18,000 residents. It's a really popular recreational site and is already home to several aquatic invasive species, including Eurasian water milfoil, Asian clams, curly-leaf pondweed, and purple loosestrife, just to name a few. In contrast, Lake Samish is a much smaller lake. It has a surface area of around 814 acres. It's an open, multiple-use lake, has a year-round kokanee fishery, and is again a source of drinking water for many of the lakeside residents. It's a popular recreational site for visitors and residents, and is currently home to two aquatic invasive species, the fragrant water lily and Asian clams, which we discovered there last year. In terms of our program development, we started out by doing a risk assessment back in 2011. We followed that by passing some local regulations and starting with a voluntary watercraft inspection program. So that allowed us to get a lot of the data and information we needed to then develop the mandatory watercraft inspection program, which we started back in 2013. And then we've revised it several times since then. In terms of the risk assessment elements we considered, we looked at the amount of recreational activity occurring on the lake. So we needed some data on boat traffic as well as access points. We also looked at the suitability of the water body to support the establishment of AIS. So in that case, we're talking about the water chemistry and how likely those species are to survive in that environment. We looked at the current distribution of aquatic invasive species and their proximity to the lake. So obviously we're getting new infestations getting closer depending on the species of concern. You also want to take into account if there are local infestations you're concerned about. The potential impacts and mitigation costs that could result from an infestation. So in my case, we're talking about drinking water sources. So those impacts are very high. Those costs would be very high if we were to get an infestation. And then looking at that existing level of protection. So do you know where these boats are coming from? Are they coming from high-risk areas? Do you have an inspection program in place? Do you have an education program in place so people know the clean, drain, dry message? So these were things we considered as we developed our program. So in terms of our local ordinances, we do prohibit the transport or release of aquatic invasive species into our local lakes. We require inspections and permits for all watercraft operating on Lake Whatcom and Lake Samish. And then this program is enforced by the Whatcom County Sheriff's Office on the water. So he's out there looking for their stickers on the boats. We do charge fees for our aquatic invasive species permits, and these fees do then go back into helping to fund our program. There are a variety of different permit options, from annual to different day passes, and then the fees do range from low for some of those lower risk boats that are easier to inspect all the way up to registered boats that are much more complex. We offer inspections at a variety of different locations at the two lakes. While our season mostly runs from April through September, we do offer inspections outside of that season at our Nevada Street location. And then during the season, we change our hours of operation at sites depending on the amount of traffic that we see there. So Bloedel Donovan and our Samish Fish and Wildlife site, we end up operating them 7 days a week from dawn to dusk because those are our highest traffic sites. In contrast, our Sudden Valley and South Bay locations are just open on weekends because we see a lot less boat traffic at those sites. Our inspection program is mandatory, and so as part of that, we do a vessel history survey and risk assessment. So we ask the boater where was the last place they had the boat in the water and when, and that helps us to determine the level of risk that boat poses to our local lake. In addition to doing that, we also do a visual and physical inspection. So we're looking to make sure there's no mud, water, plants, or animals on board the watercraft. We make sure it's clean, drained, and dried, and then we decontaminate if needed. So that could be anything from removing plants to pumping out bilge compartments to doing a full decontamination if necessary. We treat each interaction with the boater as an educational opportunity because we want to make sure they can inspect their own boat even when inspectors are not present. We collect all of our data electronically on mobile devices in the field using a database that we developed in-house. It allows us to see all their inspection, seal, and permit history in real time across all of our sites. So it's been made much more efficient for our inspectors and for our boaters. We had to change a lot of our procedures this year in response to COVID-19. So we had new inspection and safety protocols. We had to assign roles to try to minimize cross-contamination, wear PPE, socially distance as much as possible. We moved our payment system completely online this year, had to do all of our training online as well, which was quite challenging. And we ended up with a delayed start because of that, but we still ended up with increased traffic at all of our sites this year, which was really interesting. We did not have any Canadians come through our check stations this year, which was really different since we were so close to the border. And then we did also have some outreach challenges. You know, it's a big part of our program, but in an age when we're trying to really minimize our interactions with the public, and the time we're spending with the public, we had to just focus on some of those real core messages this year. Here you can see the annual total number of watercraft inspections that we've conducted each year since we started the program back in 2013. You'll see a big jump from 2013 to 2014, and that's because we added those non-motorized hand-carried watercraft to the program at that time. The blue bars represent the number of inspections conducted at Lake Whatcom, and the yellow represent the inspections conducted at Lake Samish. We increased our hours of operation and added new sites over that time, which accounts for much of the increases. However, this year was quite an anomaly. We had a delayed start, we had no Canadian traffic, and we still saw a huge increase in the number of inspections. In terms of the highlights, we had 19 inspectors working across all 4 launches. We conducted over 14,600 inspections across all 4 sites and interacted with an additional 1,100 visitors as well this year. We had 970 boats that required additional attention because they were carrying standing water that had to be removed, as well as 231 boats with vegetation that had to be removed before those boats could launch. And then we did an additional 6 full decontaminations of high-risk watercraft. This year, over 60% of our inspections occurred at Bloedel Donovan, and we did over 8,100 inspections at that site, and then we did over 3,800 inspections at the Lake Samish boat launch as well. In addition to asking our boaters where they last had their boat in the water, we also like to find out the full water body history of where that boat has ever gone in the past. That gives us that big picture risk assessment of where all our boats are going and coming from and things like that. And so this year we found out our boaters had taken their boats to over 1,004 different water bodies in 52 states and provinces at some point in the past, and that included 93 mussel-infested lakes. So we often get asked what we found on the boats. And so this year, most of the boats we dealt with were actually clean, drained, and dried. However, we did have 11% of the boats we inspected that required additional attention because they either had vegetation that had to be removed or standing water that we had to drain. We often had to pull out ballast bags to drain them and things like that. And then additionally, we had the 6 boats that we had to do a full decontamination on. So when we get those high-risk boats, we have to do a full decontamination. We pull out our mobile decontamination unit, which holds up to 400 gallons of water that we can heat up to a temperature of between 120 and 140 degrees to kill any of those aquatic invasive species. So we flush out any of those systems, ballast, tanks, engines, compartments, things like that. And then we can also do an exterior flush using our high pressure wand as well to remove any invasive species if needed. And so this year we had several boats coming in from Nevada and Arizona and different areas of concern from Texas as well that we had to decontaminate. We also have a wire seal program that sort of serves as an express pass for boaters returning to the same lake. So if a boat goes to Lake Whatcom and then they decide they want to bring it back to that lake on their next visit, as they exit the lake, we can tether that boat to the trailer using wire and a uniquely numbered plunger seal. If they come back to the lake on their next visit and that seal is still intact, we know they haven't launched anywhere else, so we don't have to do a full inspection. We can just clip that seal, enter the data in our system, and let them go on their way. So this has made it much more efficient for us, as well as safer during times of COVID, so we can minimize those unnecessary interactions. But it does still allow us to communicate with boaters and do some education. We've had 1,500 boats participate in the program this year. Most of those interactions were at Bloedel Donovan, our busiest site, and again, it really helped on those super busy days. We also have an on-site watercraft inspection program. So we do require people who have boats on Lake Whatcom and Lake Samish, who even live there, to still get their boats inspected and permitted. And if they're not able to bring those boats to our check stations, we actually offer to go to their homes to do those inspections. So we'll often do large community appointments where we do several homes in one go with boats. And it's a great way for us to do some outreach with people from our own community so they really understand the efforts that we're making to prevent the spread of invasive species to their lakes and to protect their properties. We also do early detection and monitoring. So we're looking for different new species introductions. So we're always checking for those mussels. We do that in partnership with the county and the state. And then we also look at existing species. So we do a lot of shoreline monitoring, aquatic plant surveys. We're looking at the extent of these infestations, the density of the populations, and then trying to come up with different management options as well. Most of the work that my team does is looking at the Asian clam populations at Lake Whatcom and Lake Samish, but we also document different aquatic plant species that we find. And then we also were the ones that discovered the New Zealand mud snails and have continued to monitor their infestation in Lake Paddon, which is another one of our local water bodies. So education and outreach is one of the most important aspects of our program. We do much of this at our boat launches in person and at on-site appointments, but we also use a variety of other tools as well to get the message out. And so attending conferences like this is another great tool for us too. We also try to get the word out during events. And so this year we were able to attend several bass tournaments. We had a float plane fly-in at Lake Whatcom, several small paddling events, and then we also did a lot of outreach about New Zealand mud snails at Lake Paddon. So all of these events give us an opportunity to do outreach not only with people from our community, but also people visiting from outside the area, which is really important. We also have our own website, whatcomboatinspections.com, where you can learn more about our program and ways to prevent the spread of aquatic invasive species. On there, you'll find out more information about our online Aquatic Invasive Species Awareness course. This is a course we developed back in 2014 for people to learn more about aquatic invasive species and our program. There's a short quiz at the end that people can take, and if they pass it, they can get a discount code that can give them $10 off their permits. We've had over 13,000 people take this course since we launched it in 2014. The success of our program wouldn't be possible, though, if it weren't for the partnerships we've had both locally, at the state level, as well as regionally. We've learned so much from other programs in other places, sharing information and resources and things like that. And so if there's anyone out there who is interested in setting up a boat inspection program locally, please do reach out. We're happy to share any lessons learned with you. And so with that, I'll open it up for any questions.
Thank you, Teagan. That was very interesting. Quite frightening to realize that boats come from 52 different states and provinces just to Lake Whatcom and Lake Samish. We're holding questions till the end, so everyone please submit your questions online and we'll get to them at the end of the session. So next up is Ben Peterson, and he's going to talk about invasive species in King County.
Hello, I'm Ben Peterson with the King County Noxious Weed Program, and I'm going to talk to you about Ludwigia Peploides, Class A noxious weed, control that we are doing in an off-channel wetland near Seattle, Washington. So first of all, the taxonomy of Ludwigia species is still very confusing. And this is actually text from a book called Weed Control in Natural Areas of the Western U.S. First, there are 2 or 3 invasive Ludwigia species, and of the Ludwigia species, Ludwigia Peploides, the one I'm going to talk about, there are actually 3 subspecies. One is native to California, Ludwigia Peploides, subspecies Peploides, and that's the one that's actually here in Washington that's invasive. It's not native in Washington. I'm not sure about Oregon, but I think it's invasive in Oregon. I don't think it's native there. There's one that's native to South America, and there's another one that's native to large parts of the southeast United States. So telling them apart. So Ludwigia Peploides is the Class A noxious weed. That's over here on the left side of your screen. Subspecies Peploides. The main way you can tell it apart from the native Ludwigia is that it has alternate leaves. Leaves are about 1 to 9 centimeters long. Generally grows prostrate, sort of trailing along the ground. But it can sort of mound up to be about 80 centimeters tall at the most. And it grows in water that's up to 3 feet deep. And there's only one site in Washington State where this plant occurs, and that's the site here in King County near Seattle. So it's a Class A noxious weed, super high priority. Moving on, there's Ludwigia hexapetala. This is water primrose. This is a little more common weed, Class B noxious weed in Washington State, still not native. This also has alternate leaves, so both the non-native Ludwigias have alternate leaves. That's a key way you can tell them apart from the native one. This has yellow flowers also. The leaves are a lot narrower. They're up to 12 centimeters long. This doesn't grow in quite as deep water, up to 1 meter deep. And there's a bunch of it down near Portland. Well, there's a lot of it in, the Portland area in general, but there's some just north of the Columbia River in Washington State. And I think they just found a site on Orcas Island, unfortunately. And then finally, there's the native plant called water purslane, Ludwigia palustris. This has opposite leaves. So you can see the leaves right here on opposite sides of the petiole. I'm going to turn off my camera to see if I can show you the flowers. The flowers for the native one are these little tiny green flowers that grow on the leaf axils. So you probably won't ever really notice the flowers. If you come across a plant that has opposite leaves and no yellow flowers, then it's likely the native water purslane. So to reiterate, Class A weed, here circled in red. Class B weed, Ludwigia hexapetala, and the native plant. We're going to focus on Ludwigia Peploides today. So here's a picture of water purslane, the native, circled in green, and the non-native one, Class A, floating Primrose-Willow, Ludwigia Peploides, circled in red. And this is at our site, our weed site in King County. You can see the alternate leaves on the one circled in red, the non-native one, and then the opposite little leaves. They're quite a bit smaller too, the native one, but not always. So Ludwigia Peploides occurs mostly like in the Mississippi River area, the drainages that flow into that, but other places in the southeast part of the United States. That's one of the subspecies that mostly occurs there. Ludwigia Peploides, subspecies Peploides, is pretty common in California, and also some parts of Oregon and King County. In Oregon, it's a Class B weed because it's a little bit more widespread, unfortunately. So the impacts of Ludwigia Peploides. It decreases native plant diversity. Like most noxious weeds, it crowds out native plants. It reduces open water habitat. It can reduce water flow in ditches. It can lead to oxygen levels being lowered and the pH of water being lowered. It grows very quickly. Its biomass can double in 23 days. And its growth is not limited by competition, but more so by physical space. So it can really take over a lot of habitat. This is sort of a coming back population after it was sprayed the year before at our site in King County. So it has big impacts in Europe too. There's two non-native Ludwigias that are aggressive in Europe. This is one of them on the Lorry River Valley. This might be Ludwigia hexapetala, but Peploides grows in a similar habit or similar way. So it can be really invasive. It's not native in Europe, so that's a big impact there too. So the site that we have in King County is just north of Maple Valley. And this is, once again, this is the only place this plant grows in the whole state. So that's why it's super high priority for control. And it grows in unincorporated King County near the Cedar River, just about less than a mile upstream of the Cedar River along Taylor Creek. So we really don't want it to get into the Cedar River because that'd be bad news. So a history of the place where we have the plant here in King County. It was found growing on this old wetland site, part of a rural property. And it was found about a year before a restoration project was instigated. The county was bought, or the property was bought by King County, and they did a large, about 3-acre wetland restoration project. Actually, more than 3 acres, but about 3 acres of it was created pools. Unfortunately, the county weed program wasn't able to eradicate the plant before the wetland restoration was done, so the weed has persisted. We've worked on it every year since. But as you can see through these air photos, before the restoration project happened, the plant occurred in a small wetland, like in those trees there, and then there was a bit more habitat for it to grow, unfortunately, and it still persists within that sort of pink boundary. That's sort of the full extent of where it has occurred. So here's a picture of it. You can sort of see the plants just really filling out the water there. That's from several years ago. Luckily, it's not that bad right now. The water's anywhere from like just, a few inches deep to up to more than 5 feet deep in some places. So it grows just fine there, unfortunately. Here's another picture of the plant being held. You can sort of see the scale of the leaves. So it has these sort of veins that are a little more visible, so almost like willow leaves. I think that's where it gets its common name. So first of all, getting around the site is tricky. We used to wear knee boots and hip waders, and then we started wearing chest waders. You can get in a little deeper. And then maybe 5 years ago or 6 years ago, I tried wearing a dry suit and a life jacket and bobbing around in 5 foot water, 5 foot deep water. So I was able to get to more plants that we couldn't reach otherwise. And then we thought, why don't we just shove our canoe through the woods? So we drug it maybe 100 feet through the woods or 200 feet through the woods and got to the wetland. It's a lot of canoe shoving, but it works really well, and we're getting pretty good at it, and helps to have a durable canoe. But this makes getting to a lot of the plants that are just out there in the middle of the water that we wouldn't be able to get to otherwise. And also it makes it so we don't disturb them so much and break them up with our boots. So we had to get a little creative. In terms of control work, what we did for about 12 or 13 years is we sprayed the plants every year. And I'll show you in a few slides what we use, but it's mostly foliar spray of aquatic herbicide approved by the State Department of Ecology. Often use blue dye, and of course we use aquatic surfactants to make sure the herbicide sticks. But a little tricky to get to these places because you don't want to wash the herbicide off the water. Here's my supervisor, my program manager, Steve, spraying some weeds a few years ago. Don't fall off the log. And then about 3 years ago, we got the weeds down to a small enough level that we were thinking, okay, let's just go out there and hand pull. Let's just see if we can really reduce them further by hand pulling. So we decided to go out 3 times a year and look around really carefully and hand pull. So you end up accumulating some biomass, that's for sure, but it worked out pretty well. You just have to pull really slowly to get them all, bag them up, put them in the garbage, not the green waste. So here's a sort of a chart that shows the timeline. Along the bottom is the year that the plants were surveyed and controlled, and then how many square feet of the plants. Sometimes the area was, very large, but then a very low density. So we just multiplied the area times percent cover. Sometimes it was a pretty big area and a pretty dense cover. The first maybe 8 years or so, we used triclopyr, TEA, as the treatment. And then for 2 years, we used imazapyr. And then the last 3 years of spraying, we used imazamox, which seemed to work the best because that sort of knocked it back down to a really low level, just like maybe about 100 square feet of dense plants in 2007. After that treatment, it's been a very low level. This year, there was just 25 square feet at about 100% cover, scattered around in 3 spots. So we worked a lot, and we got it down to a really low level. This graph, this shows how the plant has been scattered around the site over the years. The dots vary by year, but even just this most recent year, these red dots here, these 5 or 6 red dots represent where we've seen the plant most recently, and it's still in many different places. So that, I guess that shows how well you need to look around. And we're wondering, how did this plant get around to so many different places? So we thought, maybe it was beavers. We're not totally certain, but we did find one plant growing off by itself, just floating in the water, and it had at least 3 stems that were like sort of severed. You can sort of see these red circles around the stems. It almost looks like, they're cut. This wouldn't occur naturally. It's not like a really windy spot. There's no current or anything like that. So we thought that might be what happened. There's definitely beaver activity out there. There's beaver dams along the creek channel that's adjacent to the site, and you can see beaver activity in these trees. So that might be what happened. And this just shows how carefully you need to look for plants. This is the one plant we found during our June survey this year. Just one little plant hiding under some iris on the edge of the pond. And this other map here shows our track logs. It shows how we walked all around the site during our 2020 surveys looking for plants. So you need to be super thorough. In summary, persistence pays off. We haven't seen any movement downstream. If you have a site like this, a situation like this, search your site multiple times for growing season. That way you can make sure you don't miss anything. And search very, very thoroughly. Budget time for a very thorough search so you won't be rushed, so you can do a good job, so you make sure nothing escapes. And experiment with field control, field equipment to access your site and control methods. That's it. Here's my contact info, King County Noxious Weeds. Thank you very much.
Thank you for your very important work, Ben. And so now for our next and final talk for this session, we have high diver assisted suction harvesting. And this is by Garrett Remholt. Hi, we're having some technical difficulties and Garrett is going to be giving a live presentation as soon as the slides are make it to UW-Tacoma. So while we're waiting, we're going to start with questions. And for Julian, we have a question. Are the location data freely available from the app developers?
Yeah, thanks for that question. And that's obviously a sticking point, right? So just to be clear, people either opt in or opt out, depending upon the social media platform with respect to whether their location is actually shared. All of the Flickr data is freely available on a public API, so application programming interface in which the public can actually just query and get that information. It's obviously all anonymous, so we don't know who the people are or any of their demographic information. But it varies greatly. Firewalls, particularly around kind of Facebook and Twitter, although they have APIs in which you can access the data, it's a little bit more of a trickier exercise. So yeah, I'm on video here. Everything's above board, no big brother here. Yeah, we're accessing data much like anyone else can.
Okay, a follow-up question is on the iBobber. What other information would an angler get from an iBobber? So what's the primary use for that tool?
Sure. So the iBobber is publicly available. ReelSonar created it. It's a Seattle-based company. You can buy it on Amazon for I think around $100, and there's a lot of knockoffs. Essentially, anglers use it. It's a sonar enabled, so they can cast it and get a quick estimate of depth and bathymetry. It also has a fish finding feature. Anything above 15 centimeters, I believe, is pinged back, and it all goes then just to a phone app. It also records things like temperature and stuff like that, although that's typically not used by the angler. So for our purposes, we like it because it kind of basically pings up every 15 seconds to the cloud so we can get their location.
Well, thank you, Julian. So we have a question for Teagan. Was the increase in boaters this past summer mostly an increase from local people?
That's a good question. We did see a lot of local people out using the water bodies, which makes sense with the COVID restrictions and people needing to stay relatively close to home. But we also noticed a lot of people were buying boats from out of state and coming through. So the level of high risk boats that we had was still relatively high. Yeah, I think in terms of last water bodies, we had, they'd been in 290 different water bodies in 29 different states right before coming to our local lakes. And so that's still on par with what we had, last year. So we're still getting those, out of state high risk boats coming through.
We had another question for you. how many penalties do you typically send out every year? Like, how many tickets, basically?
Yeah, that's a good question. So on our end, we're more focusing on educating folks and making sure that they understand what they need to do to comply, and then we work with them to make sure they can pass those inspections. So on the inspection end, we get people through the process, and so they're not posing a risk to our local lakes. But in terms of the permit requirements, that's enforced more by the sheriff's office on the water. And again, they focus much more on educating people and giving warnings. This year, we actually had a much more stronger partnership with the sheriff's office. And so I haven't actually gotten the report yet from this year to know how that went, but I know they made a lot of contacts with folks. So, yeah.
Really good work up there. And we had a question for Ben. Where do you find out what herbicides are allowed for use in water? Also, do you think seedlings could still be sprouting?
Can you hear me?
Yeah.
Okay. I heard the first question. Where do you find out what pesticides are allowed in water? The Washington State Department of Ecology runs the permit program for in-water herbicide use. And they have two permits, the aquatic noxious weed permit and the aquatic plant and algae management permit. And on those permits, they list 8 or 10 or so different herbicides that are allowed for use in water under that permit. And it varies by states. Washington is a little more stringent than other states, but you need to have the permit and also a special license. And then what was the other question? Something about seeds?
The second question had to do you think seedlings could still be sprouting after your treatment?
Oh, could they be sprouting after we do the control work? Sorry, audio.
Exactly. I think that's what the question is. Yes.
Yeah. I mean, I think that could be the case, especially with the spraying. So that's why we go back 3 times a year. We go back and we go in June and then August and then again in September. So we try to be super thorough and try to find little seedlings. I think it mostly reproduces vegetatively, although the seeds are viable.
Okay, so checking in with Mark, are we ready to go to the last presentation?
Okay, great. Can everyone see that slide entitled Advantages of Dash? Are we good to go? Okay. So the advantages of Dash. This is where I want to kind of pull everything together with the high selectivity portion of the PowerPoint. And so by high selectivity, we mean that target species can be removed almost surgically. So should we have for example, a milfoil plant surrounded by native species, that individual plant can be removed just like a hand pull methodology. And that I think is a really powerful tool. Like I said in the previous slide, not having to worry about this spreading of fragments is pretty crucial because the diver can just manipulate the suction head. It's a herbicide-free method, and we think that's pretty important because in our experience with local lake communities, herbicide, although it is effective, it can be highly controversial, and the optics of herbicide use are not the best. And I think that it's a combination of public perception and a lack of education, but nonetheless, it's bypassing the negative optics of herbicide is great. And then piggybacking off of that, avoiding any sort of oxygen deficiency or suboxic conditions that can result because of a herbicide treatment is pretty crucial to prevent the chemical cycling of a lake from being drastically altered. If a herbicide treatment is conducted and the dead and decaying biomass isn't properly removed, then suboxic or anoxic conditions can occur. And another big advantage of DASH is that it can be used during all stages of infestation or invasion. And I think that's pretty exciting. It can be used right when a species is discovered, or it can be used during the long-term management. Just because it unlike diver hand pulling, it's mechanized. It can be used on a much broader, larger scale, which is a pretty cool thing. And then also, DASH is suitable for sensitive bodies of water, like Whatcom, so drinking water reservoirs, sensitive watersheds, bodies of water where a herbicide or biocide application is not appropriate. DASH can be a viable alternative to herbicide in that respect. So if we just advance the slide to the the disadvantages of is it the disadvantages of DASH? Is that the next one, or it looks like we just have standard? There we go. So the disadvantages of DASH. I think, like any methodology, there's always some drawbacks. I think that the big initial drawback is the fact that DASH is fairly expensive, especially relative to chemical treatment. Putting any human underwater has inherent associated costs with it. And then on top of that, generally one treatment isn't always enough. Therefore, DASH is a more time and energy intensive process than, say, the application of herbicide. Especially some herbicides are extremely effective. But that's not to say that that time and energy is not worthwhile. Also, the rate of progress decreases as density of the target species increases. So if there's more milfoil or there's more hydrilla, then the rate of progress of removal of that plant holistically will decrease. And also the rate of progress and therefore the efficiency is very much dependent on the crew operating the equipment. So if a diver or divers are not properly trained in DASH or do not have much experience, then there will be a decrease in efficiency. Also permitting has been an issue for us in that because this is such a new technique being used on the West Coast. Fish and Wildlife has not really been sure how to permit this. We've proceeded by just acquiring standard HPAs, but we're still working with Fish and Wildlife to streamline this process, and that's been a sticking point for us. So if you could advance to the next slide, the standard DASH filtration system. So most DASH systems, most DASH boats that are commercially available, utilize just a simple direct bagging or a simple screen filtration mechanism to sieve out or separate biomass from the water stream or the slurry. And this simple methodology, though it makes it really easy for the operators, like the direct bagging, was not adequate for operation in Lake Sammamish and Lake Washington, which are the primary lakes that we operate in. And that's due to WDFW's regulations to protect endangered sunlighted species. So what we had to do was devise our own system to meet those unique requirements. And so what we did was that we developed a wet well catchment combined with a sorting table, and then also combined with a turbidity containment and management system, so that if we did mobilize any bed material, it could be and can be directly redeposited to the lake bed. So on the picture to the left here of your screen, that's just an example of the direct bagging system, where you have the end of the discharge just puts the biomass and slurry into the bag, and then whatever isn't caught by the bag, so whatever fragments aren't caught by the bag, are redeposited back into the water body. And that kind of defeats the purpose of DASH for early maintenance, because you're essentially going through a field of dandelions, just kicking them. So you're not really collecting all the fine fragments, and thereby you run the risk of actually propagating these invasive species. And then in the picture to the right, that is just a really basic, a simple screen filtration mechanism where the slurry is deposited onto the screen, and then the biomass is dewatered, and then the biomass can be removed and then properly bagged. Generally, those simple screens aren't adequate enough because the orifice size of the screen is not fine enough to really catch all of the reproductively viable fragments. So ensuring that a DASH system has a fine filtration screen or a secondary screen is pretty crucial. So if you could go to the next slide, biomass management system. Let's see, I think you went one too far. There we go. So the biomass management system, and this is what we came up with as our method. So we built this system to meet all the unique requirements set forth by Fish and Wildlife, and it's broken down into three primary stages. You have the gross biomass separation, you have the stratification phase, and then you have the fragmentation collection, and that is just part of the filtration mechanism. And then below that is the actual turbidity and sediment control system. So like I said, the triple stage filtration is pretty crucial because it allows us to catch all sorts of fragment sizes. So the gross biomass separation will collect a large ball of weed biomass, say just a large clump of milfoil. The water slurry will allow the water to fall through into the wet well catchment. The wet well is important because should we suck up a non-target species like a fish or any sort of invertebrate, the survivability of that environment is much, much higher, so that they are in fact in water and capable of sustaining life. And then the third point, which is the fine fragmentation collection, we have a filter screen that we can change the orifice size depending on the target species. And so say we're removing milfoil, then we can use a very fine filter mesh. If we're going after Asian clams, we can use a larger orifice size. So that interchangeability of the finer fragmentation screen is pretty important. So if you could go to the next slide. This is the biomass management system, turbidity management. Actually, the DASH plus BMS, which is our biomass system. So this is, I wanted to include these pictures just to take a step back and look at the whole system from afar, and why we think that DASH is a really great tool for the management of invasive species. So I think you may have advanced the slide just one too far. You could go back to the DASH plus BMS. There we go. Thank you. So on the picture to the left, these are all the stages of the filtration system. You have stage one, which is the gross biomass separation. Stage two, which is the stratification stage, and that's where fragments can stratify out of the fluid stream and then be collected by the third stage, or the fine filter. So the transfer chute is actually how we transfer the biomass to our biomass skip. And we found that to be extremely effective, and it allows us to holistically get rid of bags, because we were concerned that bagging any of the biomass leaves an opportunity to inadvertently capture a non-target species and then forget about them. And that's, working in a body of water where we might be interfacing with highly threatened species. It was of utmost concern, and it was an utmost concern of ours to make sure that we didn't jeopardize these species at all. So the picture in the middle, that's just the biomass skiff underneath our crane, and that's just lifting I think it's a 2,000 pound bag of milfoil. But I think what's really cool, and it's highlighted by this picture on the right, is that's a 6,000 pound pile of Brazilian Elodea that we were able to remove in a day from Lake Sammamish. And when I say remove, I mean, root crown, stem, fragments, everything holistically was removed. And so I think that picture really highlights the efficacy of DASH for a later stage infestation as a management tool. So most of diver handling activities are done in the early stages of an infestation. But I think that this picture really underscores how DASH can be a tool utilized in the latter half of an infestation, where we're just managing and conducting mitigation as opposed to remediation. Or ideally, with enough time, that mitigation would become remediation. So if you could advance to the biomass management system, I want to just kind of underscore the turbidity control system. So controlling turbidity is really, really crucial. And that was one of the major concerns that Fish and Wildlife had, and we had as well. So what we did is we affixed a collapsible, flexible piece of ducting to our discharge ports on the hull of our pontoon boat, and that would consolidate and redeposit any collected bed material, which we rarely actually move or mobilize bed material, but to make sure that if we had to, we were able to just redeposit that to the lake bed. Most DASH operations don't actually generate a whole lot of turbidity, but knowing that a diver, should they need to mobilize some bed material to ensure that they completely remove the root crown of the target species, should it be a plant, it is nice to know, because then we can really reduce the variability associated with diver hand removal, just so that we know each plant is completely removed, and it's a good deal. So low species specificity, this is the second part to the title that I think is really cool, is that if you were to take DASH and couple it with a proper biomass control system or filtration system, there aren't very many species that you can't target. So in terms of plants, we can target immersed, floating, or submerged plants. We can also use them for invertebrates. So when we start talking about how do we control New Zealand mud snails, how do we control various invasive mussel species like zebra mussels, using DASH is a very promising methodology. We've been able to conduct a few tests utilizing rice grains to simulate New Zealand mud snails, and we put them on the lake bed, sucked them up, and then counted all the grains of rice in the catchment. And we put 30 down, and we sucked 30 up. So I think when we start looking at these large scale invasions of invasive species, knowing that we can use a tool that's not necessarily a chemical tool, I think is really important, because it gives us a whole new methodology to control invasive species, which are only going to get worse. I mean, as globalization increases, as waterborne commerce increases, these invasive species are only going to be spreading faster. And so knowing that we have more tools in the toolkit to control these is really important. And so just to kind of sum it all up for the low species specificity, if it lives underwater and can fit through the suction head, we can remove it. And I think that's a very powerful thing. So I think that's all I really have, and I'm happy to take any questions. I also want to thank the WALPA hosts for putting this whole presentation together. It was great.
Thank you. Thank you, Garrett. Can you hear me?
Yes, I can hear you.
Just checking. Yeah, thank you for being flexible on bouncing over to a live presentation when you had such a great presentation pre-recorded. So we had one question for you, and that was, how do costs compare with diver-assisted suction harvesting to the big mechanical harvesters?
They can actually be less expensive. Operating those large surface-borne rototillers can be extremely expensive. Logistically, the challenges with finding someone who either owns or rents one has been pretty difficult. And because of their size, they're also really hard to deploy in certain bodies of water. So we found that Dash can actually be cheaper than a surface rototiller. And I think, I can't give you any specific metrics on that because it kind of depends on who has the surface harvesting device. But it's definitely comparable, if not cheaper.
And are you working all across Washington, or do you have many sites in Washington at this point?
We do. We've worked with Snohomish County, the surface water management team up in Lake Goodwin and Lake Shoecraft, and also Lake Washington and Lake Sammamish, Liberty Lake down in Tacoma. So we can work all across Washington, and so we're not particularly limited by geography.
Well, thank you.
Thank
you. We're going to circle back to Teagan with a couple of questions. And just how big is your team, Teagan?
Yeah, so this year we had, so I'm a full-time employee, and I have one other full-time employee, and then I have 2 9-month seasonal staff, and then we had 17 6-month seasonals this year. But in the early days, we started with 3 people and 8 people, so we've been growing steadily over time.
Then do you coordinate with other counties, or do you, like Snohomish, Skagit, Island County, do they also have programs like this, or?
Yeah, that's a good question. We definitely are open to working with other places, and so I do get calls from other folks. So I've gone out to Lake Chelan to talk to their board about potentially setting up a program. But no, we're very unique in Washington State in terms of this local effort. But we do coordinate mostly with our own, programs within our own county. When we find infestations and things like that, we'll check in with other places, and then we do attend a lot of regional meetings and state meetings and work closely with Fish and Wildlife, because they also have inspection stations on our borders as well.
And this is a question I had as well as someone else had, was how do you control float plains as invasive species vectors?
That's a great question. So we actually work really closely with the Washington Seaplane Pilots Association. And so they do a lot of outreach to their membership on ways to prevent the spread of invasive species. And then when they come in here, for example, they send us a lot of their logs and information. So we know where they've been launching and kind of can assess their risk. But for many of the planes, if they are true float planes, they are coming from one lake and having to land. So it's difficult to inspect them before they land. In the case of the event we had this summer, though, most of those planes were amphibious. And so they actually had come from airports on land prior to coming to Lake Whatcom. So those are the kinds of practices we encourage, so they're not, transporting any species between lakes.
Yeah, that sounds good. It looked kind of funny with the big airplane coming in. You just don't know. Thank you. So we have a question for Ben. How do you think that plant originally got into the watershed or the wetland?
Good question. I was just talking with the restoration site manager, and they definitely knew the weed was there before they started the project. And he said it looked like the pond, or it was like an area that looked like had aquarium rocks and things like that. So it could be that it came from an aquarium dump from the previous landowner. We don't know. Yeah, I wish we knew. Yeah.
It looked pretty extensive in your pictures. pretty well established at the beginning.
Yeah, it was really scattered around because the site had a lot of woody debris and overhanging willows and stuff. So it takes a lot of work to really look carefully.
And then we have a question for any one of the panelists. On average, when a new invasive species is discovered in a lake, river, or reservoir, where on the invasion curve is this species, i.e. early to well established? And how does this play into the choice of how to manage it or not? So I guess we'll go with you first, Ben.
I mean, if it's an uncommon species, a Class A weed, such as this Ludwigia, then it would be super high priority to get rid of. In general, we know all the lakes that have the common weeds, such as milfoil and Brazilian alidea. But yeah, we're definitely, our main focus would be early detection rapid response.
Teagan, anything?
Yeah, I think, for us, it's dependent on the species. I do think if you have these early detection programs in place, that's really important to be able to catch them early. In the case of when we found the Asian clams in Lake Whatcom, it was actually residents that found them on a beach. So they had been present for several years by the time we found them. And unfortunately, that's a common story. So I definitely think having those early detection programs, getting out there, surveying, and working with your communities, if you have people living on the lake, so that they can ideally catch these things, is really important.
How about you, Julian?
No, I think they covered it.
Covered it okay?
Yeah.
All right. And then just a final question here for Garrett. It kind of relates to the previous question. Can you estimate the cost for a treatment with the DASH approach?
I can, and I think it depends on the size of area. The larger the treatment area, the less expensive it is, just because once our boat is deployed, leaving it there and being able to conduct operations in a discrete location makes us more efficient, and therefore less expensive. So most of our unfortunately we only really work with private homeowners for the most part right now. We've been working towards reaching out to municipalities, government agencies, statewide agencies to try and say, hey, this is a new management technique. But for now, we generally charge on a square foot basis at about 40 cents a square foot. But that also depends on growth density. That's assuming maximum growth density, so a plant every 6 to 12 inches. But as the density decreases, we can move faster. A great example of that is our work up with Snohomish County on Lake Goodwin, where that was more of just a survey where we actually weren't really using the dredges. We were just diving on scooters looking for invasive milfoil, marking the location of each plant, and then removing them. And the cluster of plants was fairly large. Then we deploy the dredge. So I think it'll kind of depend. But generally, for a good estimate is anywhere from $2,500 to $3,000 a day is a decent metric.
Well, thank you for that estimate, and thank all of our panelists today for this very interesting and important topic. We're done with questions, and we'll be meeting again back up for session 7 on waves and source tracking at 1:20. 1:20. We'll see you again.